All right. Woo! Okay, moving on in our unit on eukaryotic cells. So remember last time we talked about the nucleus, the double membrane of the nucleus, nuclear pores, chromatin, all of those things, nucleolus, mRNA moving in and out through the nuclear pores, why eukaryotic cells are more efficient because of compartmentalization. So we're going to talk about some more of these compartments today. Today we're going to be talking about the endomembranous system. Endomembranous system. Basically, it's a system of membranes on the inside of the cell. Endomembranous system. There you go. And the endomembranous system, the main big component of the endomembranous system is called the endoplasmic reticulum. You may have heard of it, the endoplasmic reticulum. There's rough endoplasmic reticulum and smooth endoplasmic reticulum. And so the endoplasmic reticulum has 3 domains. That just means areas, 3 areas where it works. And, well, let's get into it. Let's talk about it. So let's show you an overview of the endoplasmic reticulum and its 3 domains. So this is it. So you can see there's the nucleus in the center, and that there, let's see if we can make that bigger. That's the nuclear membrane, but just the outer nuclear membrane. So remember, the nucleus is like double membraned. It's got 2 lipid bilayers with a little perinuclear space. That's the space in between the 2 membranes. And the inner nuclear membrane, which surrounds the chromatin, is anchored to the outer nuclear membrane with those nuclear pores, those things that let stuff through, those complexes of enzymes. The outer nuclear membrane, so there's the inner one, then there's the perinuclear space, and then there's the outer nuclear membrane. The outer nuclear membrane is the first domain of the endomembranous system. So the first domain is the outer nuclear membrane. And if you remember from our diagram yesterday, we didn't really go over, but that has this. So this is it. This is the outer nuclear membrane. So that's the first domain of the endomembranous system. There's the perinuclear space, and that's the inside. There's the inner nuclear membrane, those laminar proteins, and the inside of the nucleus. And you can see there's ribosomes dotted on here. Ribosomes are the site of protein synthesis. That's where proteins are made, protein synthesis. So information, mRNA from the nucleus comes out, it goes into a ribosome, and it gets translated into amino acids. And so remember proteins, so the enzymes and proteins, this is where they're made. So it could start right there, right on the outside of the nucleus on the outer nuclear membrane. So that's the first domain. And so you can see it's connected to the perinuclear space. So you could actually go right into the perinuclear space. And what happens is the perinuclear space actually opens up into the second domain. See, there's the perinuclear space, and it opens up into the second domain, which is the rough endoplasmic reticulum. And the advantage of having all the ribosomes and stuff right there is if you need to make something really fast, you can make mRNA inside the nucleus, and right there are ribosomes to right there make a whole bunch to make protein and send it out, or send it into the nucleus. If you need to do something in the nucleus, you're doing cell division, for example. If you're doing cell division, you're going to have to copy all of the DNA in there, and you're going to have microtubules, which are proteins that are going to divide the cell and do a whole bunch of stuff. You can do it right there. It's all right there. So that's the advantage. You got direct access to the nucleus by having the first domain and ribosomes right there. So that's the first domain. The second domain you may have heard about is the rough endoplasmic reticulum. And why is it called the rough endoplasmic reticulum? Because it looks rough. Look, this is what it looks like under an electron microscope. It looks really rough. And so what you're looking at here is those lines are membrane, and the little dots are ribosomes. Lots and lots and lots and lots of ribosomes. And so ribosomes are the site of protein synthesis. It's where proteins and enzymes are made. So this is an overview again, and this is the rough this is the area of the rough endoplasmic reticulum. And so it's full of ribosomes. Dotted, that membrane is full of ribosomes. And look, they're all interconnected. And the spaces inside, these are called cisterns. These are all interconnected, and they're called cisterns, those spaces inside. Cisternae. And so in here, proteins are made. Now, they're not fully made. What will happen is they'll just get translated into peptide chains. So amino acids will get put together, and maybe you'll get this is where the first level of protein structure is. Remember we talked about protein structure, the different levels, first, second, third, fourth. So you'll get first and second here maybe. Stuff will start to be made in here, but you're not likely to get third, and definitely not to get quaternary, fourth level of structure of proteins here. So you're getting the peptide chains, you're getting the order of peptides made, and then what happens is once they're made in here, the edges, the membrane. So you can see this is a double membrane again. This is a lipid bilayer that just goes on and on and on and on and on and on. And these stacks here are cisternae. And what happens is these bleb off. That's the actual term. It's called blebbing. You can see some blebbing going on here. So there's the stacks. There's probably the cistern, the inside area, and stuff is being made in there. And then what happens is it'll pinch off. The membrane will pinch off, and that's called blebbing. And a little vesicle will go off with the proteins inside it, with the peptide chains inside it. That primary, secondary structure will be inside those little things, and it's blebbing off. And those are called transitional vesicles, and we'll talk about where they go in a second. So we got transitional vesicles blebbing off, full of immature protein. Unless you're making microtubules where you're making those proteins that only need secondary structure, then they bleb off and they can go be microtubules somewhere. But for now, that's the second domain. It's the site of protein synthesis, and it's dotted with ribosomes. It's got lots of ribosomes, and the membrane is folded, appears like flattened discs. These discs are called cisterns. And vesicles bleb off, that's the term, bleb off the cisterns of the ER, and they go off. We're going to talk about where they go. The third domain is called the smooth endoplasmic reticulum, because there are no ribosomes. No ribosomes in the smooth. So it's smooth. It's not dotted with ribosomes. So that's not where proteins are made. No ribosomes, you can't make proteins. No protein synthesis. No ribosome, no protein synthesis. So let's go back to our little overview. So look, this is all continuous. It's all connected. So first domain, the outer membrane of the nucleus and the perinuclear space. Then you got the second domain is connected right to it is the rough endoplasmic reticulum, and then connected right to that is the smooth endoplasmic reticulum. And you got to imagine this is all one big 3D. This is just like it's sliced in half, but it would go all encircling, all inside the cell. Some cells that have to make a lot of protein will be basically all full of just rough endoplasmic reticulum. Will be just a huge thing of rough endoplasmic reticulum. Okay, but let's talk about smooth endoplasmic reticulum. So you can see the cistern have a different shape. They're not these flattened discs, they're tubules. They're these tubes. They got a round shape, and there's no ribosomes. And what happens in here, what is made in here, are lipid or oil, fat-based things are made inside the smooth endoplasmic reticulum. That's what it looks like under an electron microscope. You can see that's the slice through all those tubes. And so what's made in here are hormones. Hormones are fats. And what hormones do is hormones go and they control which parts of DNA get expressed. So they're very powerful. They're very important because they control which enzymes are made or which proteins are made. So that's hormones. Cholesterol is made in here. And you remember cholesterol is responsible for making the lipid bilayer, that phospholipid bilayer, making that rigid. So less cholesterol, more cholesterol, the lipid bilayer, if there's more, it becomes rigid. That cholesterol fits in between those phospholipids and strength, so the bilayer isn't all fluidy. So it's very important. If you've got a lot of smooth endoplasmic reticulum, you're probably making a lot of cholesterol. You're in an area where cells need to be really rigid. And then the last thing, huge important thing, is phospholipids, right? Phospholipids are those things that make up the cell membrane. So they're the main component of the cell membrane. Remember, we learned about cell membranes is you have phospholipids. They're the ones with the tails that are nonpolar, and the heads that are polar, and basically huge important thing. So you're making all of that. So you're making these things. The other important thing that the smooth endoplasmic reticulum does is, because as your cell is working, these things, these oily things and bits, they're sort of flaking off, and the cytoplasm is getting full. Like the inside of the cell has all this junk in it, basically like stray phospholipids, pieces of fatty acid tail. This smooth endoplasmic reticulum takes all those things in, so it cleans the cytoplasm and then repackages them, rebuilds them into hormones, cholesterol, and phospholipids. And that's going to be enzymes and proteins in here doing that, controlling that, and making those, putting those phospholipids. Okay, so those are the three domains of the endoplasmic reticulum of the endomembranous system. Okay, first three the only three domains are the ER. So the endoplasmic reticulum is composed of the first domain, which is the perinuclear space and the outer membrane of the nucleus, of the nuclear membrane. It's composed of the rough endoplasmic reticulum and the cisternae of the rough endoplasmic reticulum, where protein synthesis happens, where proteins and enzymes are made. And the third domain, which is the smooth endoplasmic reticulum, where lipid-based molecules are made. Okay? The next bit of the endomembranous system is called the Golgi apparatus. So if we look at our cell here, we could see, all right, so there's the nucleus. So there's the first domain. That's the, let's say that's the outer nuclear membrane. There's the rough endoplasmic reticulum. This cell doesn't seem to have any smooth endoplasmic reticulum, but it'd be somewhere over here if it did. And there's the Golgi. Golgi looks like this. It looks like these flat pancakes. That's the Golgi apparatus. And so these are the cisternae of the Golgi apparatus. Again, these are membranes. These are kind of membranous sacs. And they're not connected. Unlike these, which are all interconnected, these are not connected. And so let's talk about the Golgi apparatus. Okay, so let's scroll down. There's our smooth endoplasmic reticulum, and there's the Golgi. Let's see if we've got a nice image of the Golgi here. I think we do. So there we go. There's the sacs of the Golgi. Now, what happens is, oh, good, and we've got it also drawn with the smooth endoplasmic reticulum. So remember when I said, okay, that the endoplasmic reticulum, so this one doesn't seem to have a smooth endoplasmic reticulum, but this is the rough endoplasmic reticulum. And I remember when I said the rough endoplasmic reticulum, once a protein is made inside the rough endoplasmic reticulum, you've got a peptide chain, you got maybe the first and secondary structure, so it's just the order of amino acids, and then a vesicle from the rough endoplasmic reticulum blebs off, right? It pinches off, and it's full of those peptide chains. Well, what happens to those peptide chains now? Those peptide chains, they got first and secondary structure. They need to be folded and have that tertiary structure, that third level of structure. Remember, where the hydrogen bonds will just will hold it together, and if it's an enzyme, give it that specific shape. Or quaternary structure, where different peptide chains will come together and make a full enzyme, a whole big full thing. That starts to happen in the Golgi. That happens in the Golgi. So what happens is the vesicle with first and secondary structure of those proteins blebs off from the rough endoplasmic reticulum, and that vesicle is called a transitional vesicle. and it travels to the Golgi, and it travels to the cis face of the Golgi. That's what it's called. So the cis face faces the nucleus. You got to imagine the nucleus is somewhere over here, and the rough endoplasmic reticulum is over here, and the smooth endoplasmic reticulum is somewhere over here, because the same thing happens. Smooth endoplasmic reticulum also, lipid-based molecules from the smooth endoplasmic reticulum will also travel to the cis face. And what happens is the cis face, it doesn't actually exist. I know, that sounds crazy, but it doesn't exist, because those vesicles, those transitional vesicles come and they glob together and they make the cis face of the Golgi. Now another thing is coming to also make the cis face of the Golgi, and that's these little vesicles that also glob onto the cis face, and they're coming from over here, they're coming from back here, and they glob onto the cis face, they make the cis face along with the transitional vesicles from the rough endoplasmic reticulum, so those precursor peptide chains. Those vesicles glob on, and what's inside those vesicles are enzymes that will now fold the proteins and start assembling the final stages of the assembly of those proteins and enzymes, combining them together to make functional enzymes and proteins. So, and what will happen is that cis face will you've got those enzymes and proteins coming that are going to assemble the bits that are coming from the rough endoplasmic reticulum. So they gleb together, they make the cis face, and then they sort of just move along as more cis face is getting made. They move along. So the motion of these cisternae moving this way is called anterograde motion. They're basically being pushed by new stuff being made above them. They're getting pushed down. They're getting pushed out to the outside of the cell. So what happens is you've got those precursors. They come along. All the little blebbed off vesicles from the ER, they come along. Some of these enzymes, they work together and stuff. And then you've got the enzymes coming here that are going to fold, give it tertiary and quaternary structure, and they get pushed down as new stuff gets made. So they're moving in anterograde motion until they make it to here. This is the trans face of the Golgi apparatus. And what happens at the trans face is this cistern, the trans cistern, breaks apart into little vesicles, and now are fully formed proteins and enzymes. So some of those vesicles contain fully formed proteins and enzymes that are going to go off and do stuff. They're going to go off and go to the cell membrane and leave the cell and do stuff outside of the cell, or they're going to stay inside the cell, and we'll talk about that in a second. And this transphase also breaks up into the little vesicles that travel this way, that get recycled. Basically, those enzymes that have been assembling the proteins here, they get recycled to start again at the cis face and make the cis face. This movement of those vesicles, the recycling, is called retrograde motion. Retrograde. So you got anterograde is moving from cis to trans, and retrograde is moving from trans to cis of the Golgi apparatus. Okay? So if this is a cheek cell, pretend this is a cheek, this is inside a cheek, inside your cheek. So what's going to happen is a stimulus comes. You've just eaten a piece of bread. You've got to digest the starch that's in the bread. You've just bitten the bread. Your taste buds have detected bread. Oh my goodness, bread. Some kind of delicious, like French bread, like a fresh baguette. Magnifique. And so your brain has gone into action. It's sent out hormones to say, ding, 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 we're going to be digesting bread. We're going to be eating bread. Get that sugar out of the bread. We're going to eat the starches out of the bread. And so what happens, your brain sends out hormones. Those hormones travel into the nucleus. They activate a section of your DNA that codes for amylase, an enzyme that digests starch. And so now the nucleus is starting to make mRNA strands. Those are genes for the amylase enzyme. Those RNAs leave the nucleus now. The DNA has been converted, transcribed into mRNA. The mRNA goes into ribosomes in here, and it gets translated into peptide chains of amylase. So those amino acid chains of amylase, but it's not functional amylase yet. It's just still in the rough endoplasmic reticulum. It only has that primary structure, the primary structure of the enzyme. It's not all folded up yet, all complete. And so then a vesicle blebs off, a transitional vesicle blebs off. Now we've got the amino acid chain. It blebs off. It travels to the cis phase, boom, those vesicles of all those amylase peptide chains, a whole bunch of them will bleb together, will fuse together to make the cis phase. Meanwhile, retrograde vesicles come here. These vesicles contain enzymes and proteins that are going to now shape, they're going to put together finally all the different, maybe amylase is probably more than one peptide chains they will give us. So they'll give a tertiary, quaternary structure, assemble that amylase as it goes, and as they assemble it, more amylase is being made on top. They'll assemble it, finally gets to the trans phase. Amylase is all complete. Those vesicles now full of completed amylase leave. The enzymes that were forming the amylase go back in retrograde motion to the cis phase to make more. Then those vesicles exit out, and they travel to those vesicles full of amylase. They travel to the cell membrane of your cheek and get exocytosed. And amylase goes inside your mouth and starts breaking up that bread, and you start getting that delicious sugar from the bread, and it starts to go down. That's how it happens. That's the process. That's the job of the endomembranous system, is so that you can make things. So let's talk about the Golgi, and let's talk about some of these vesicles, what happens to these vesicles, and how it works. This is just an electron microscope image of the Golgi. You can see all the vesicles blebbing off. So we probably have, this is retrograde motion of those vesicles forming the cis phase, and this is the trans phase. Vesicles breaking off and going this way. It might be the other way around. It's hard to tell. I don't know where the cell membrane is on here. Okay, so there are two main types of vesicles that will come off the Golgi. So you've got, like I said, you've got those vesicles that contain those enzymes that are getting recycled. You've got those, but you also have the ones that are going to be doing stuff, that now the Golgi has assembled, finished the enzymes, the proteins that it has. And so there's two types. The first are secretory vesicles, and there's two types of those. There's ones that head for the cell membrane, like that amylase that I was talking about. They go right to the cell membrane, and they fuse with the cell membrane and get dumped out immediately. Just all gets dumped out immediately, right away. The second kind, so they go out via exocytosis. When we talked about exocytosis, most of you answered that question on the test, right? The second kind is, what happens is they go to the cell membrane in vesicles, but they don't get exocytosed. They build up at the cell membrane, and they wait for a signal to come. And when a signal comes, they all get dumped all at once. Most likely, I don't know for sure, but most likely that's actually how amylase works, is it gets built up, it gets made and built up at the edges of your cheeks, and then when there's a stimulus of sugar inside your mouth, immediately all those vesicles get dumped out and more amylase starts to get made in behind, so that you have an immediate response. So those are the two, is you get ones that get dumped out immediately and ones that wait until there's a stimulus at the end. Then the second main kind, or I guess we call it the third kind now, are vesicles that don't leave the cell, that work inside the cell. So they get made in the Golgi, and then those vesicles now are called, make sure I get it right, is endosomes. So you have these endosomes, these vesicles now inside your cell, and they're full of what's called hydrolytic enzymes. Hydrolytic enzymes do catabolism. cat. They cut stuff up. That releases energy too, remember. And so how these vesicles work, they're endosomes. Now, you don't want them cutting stuff up all the time, right away. That would be bad. That would be really bad. So those hydrolytic enzymes are in a precursor form. And basically what they have is if you guys can still see me. What they have is, so here's the active site of those vesicles, and what they have is a sugar chain that blocks the active site. So they can't be eating stuff. So that's an endosome. An endosome is full of these enzymes for chewing stuff up that are inactive. They're in a precursor form because they have this sugar blocking them. In the membrane of those endosomes are two things, something called clathrin, which is one of these peripheral proteins. Remember those proteins that sit in the cell membrane? And proton pumps. So those are integral proteins that sit through both layers of the cell membranes, and they use ATP to pump hydrogen ions, those are protons, inside into the endosome, that vesicle. And so what happens is let's say your cell, through endocytosis, so this is the outside of the cell membrane, your cell takes something in that it wants to eat. Maybe this cell is a white blood cell, and it has now consumed coronavirus. It's eaten a coronavirus that it wants to destroy. What it's going to do is that cell membrane is going to invagulate, is going to come in, and so now there's going to be a vesicle inside the cell that is got coronavirus particles in it. And now one of those endosomes is going to come in contact with this vesicle that's got full coronavirus. And when they come in contact, there's clathrin in the coronavirus vesicle, and there's clathrin in the endosome. They come together and they get activated. And that clathrin will go, hey, proton pumps. Remember the proton pumps? Those are the ones on the endosome. Turn on. And those proton pumps will turn on, and they'll start sucking in hydrogen ions into the endosome. I know this is a lot, but bear with me. When you suck in hydrogen ions, you increase the H, you lower the pH. Remember we talked about that. It becomes acidic inside the endosome. And that acidic environment, what it does is now those precursor hydrolytic enzymes, the enzymes that cut stuff up, that sugar thing that is blocking their active site, when it becomes acidic, that sugar thing it breaks off. And now those hydrolytic enzymes can eat. And the endosome fuses with that vesicle that had coronavirus in it. And this endosome, when it becomes acidic inside, it's no longer called an endosome. It's now called a lysosome. It changes names when it's acidic inside. It fuses, and now those activated hydrolytic enzymes and the acidic environment are all with those poor coronavirus molecules, and those enzymes chop the coronavirus up into little bits. That's how your cell eats. That's how the Golgi plays a part in it, by making the endosomes. So remember, clathrin is a peripheral protein that activates the proton pumps when it comes in contact with other clathrin of vesicles that have stuff that you want to eat. The other thing that also happens that these endosomes are used for is recycling old organelles. So here we've got, let's say you've got a mitochondria here. There's a mitochondria producing ATP, but it's worn out. It doesn't make very good ATP anymore. So what will happen is an endosome will come in contact with this old mitochondria, and usually what happens is also this mitochondria will get covered up in a vesicle. The two will come together. The endosome will turn on and turn into a lysosome and chop up that mitochondria. Those enzymes will come in, those hydrolytic enzymes will chop up the mitochondria, and then the leftover bits will go into the smooth endoplasmic reticulum, because basically that mitochondria is just cell membrane, and it'll get chopped up, it'll all burst, and it'll go into the smooth endoplasmic reticulum and get recycled back into new cell membrane or new organelles, whatever it needs to be. Okay, this is a huge unit. So let's just sum it up real quick. Endomembranous system, composed of the endoplasmic reticulum and the Golgi body, and those endosomes, which then turn into lysosomes. That's the endomembranous system. And the endoplasmic reticulum has three domains. The first domain is the outer nuclear membrane and the perinuclear space. The second domain is the rough endoplasmic reticulum with all those ribosomes that is responsible for making proteins or making peptide chains. It's making the first two levels of protein structure. The third domain is the smooth endoplasmic reticulum, which is responsible for making hormones, cholesterol, and phospholipids, as well as cleaning out, recycling lipid-based molecules out of the cytoplasm. That's the third domain. Then you also have the Golgi, which is those flat cisterns. The cis face gets made by transitional vesicles coming from the rough ER and the smooth ER, containing precursor to enzymes and lipid molecules. They get assembled in the Golgi. They get finalized in the Golgi. They move anterograde, while enzymes that do that assembling are moving retrograde in vesicles, coming from the trans face to the cis face. The things that are getting assembled are going from the cis face to the trans face. At the trans face, everybody breaks up into small vesicles. Either they get secretory vesicles that go to the cell membrane and they get exocytosed right away, or they go to the cell membrane and they hang out at the cell membrane and wait. They build up and they wait to get all exocytosed at once. Or from the Golgi, from that trans face, they turn into endosomes full of hydrolytic enzymes. The endosomes in their membrane have clathrin proteins, peripheral proteins, that activate proton pumps in that membrane that make the inside of the endosome acidic, that removes the sugar that is on the hydrolytic enzymes and activates them. And those endosomes then become lysosomes, and they can eat stuff for the cell or break apart and recycle old organelles. Whew. A lot of stuff in here. Okay, so the review questions are up. Do the review questions. Love you. Good luck.